There we go. All right. So thank you to the organizers for inviting me to speak today. In this workshop, we're thinking about broader impacts, and part of that is engaging with stakeholders. So I thought I'd take a minute to talk about the different categories of stakeholders. So first, there are those that are directly involved in the project, and then your funders, and even critics that might be skeptical of the project and its outcomes. There are also those that are indirectly involved but are the primary beneficiaries of the project, and these can be other researchers, trainees, or society. With the glaring injustices in this country and the recent atrocities against Black Americans, it's especially important for us to be intentional about involving non-dominant populations at all levels. In our conversations today, I hope we can pause to reflect on our efforts to engage minority stakeholders and think of ways going forward how we can be more deliberate in how we approach equity and inclusion. So in my next few slides, I'm going to give you some examples of my own experiences interacting with the different types of stakeholders and then discuss my perspective as a stakeholder in the Plant Cell Atlas. So I'm the director of a project called Crops in Silico, and the central thesis of our project is that we can use integrative and multi-scale modeling to build accurate representations of crops. There are thousands of mathematical models of plants that have been published, and even more studies with empirical data that can be used to parameterize and test these models. And we can think of these models as puzzle pieces that when put together, they make up the full picture of a plant system. So we just need a way to put these together. In an effort to do that, Crops in Silico has three major components. The first is a computational framework, which is the messaging system. Then there are the integrated models that we use to address specific biological questions. And then there's also a community component to centralize models and data. And we're working on each one of these components with our stakeholders in mind. So first, there is the computational framework that is the glue that connects all of these pieces. And we want this tool to be widely used. So we've engaged with those that are both directly and indirectly involved in the project to get an idea of what are the needs and then try to meet these needs so that there's minimal burden on the user. And so the outcome of this is listed here in our current capabilities and implementations of our framework, which is called Yggdrasil. And so, for example, our capabilities is that the framework can be run on Linux, Mac OS X, and Windows. And then we've built in support to connect models that are written in Python, MATLAB, C, C++, R. And because of feedback from the community, we're also adding in capability for Fortran and SBML. Next, we aim to address specific questions that are relevant to plant biology and food security. And this is what's important to many different types of stakeholders, including researchers, society, and funders. We recently published a proof of concept where we use the Yggdrasil framework to integrate models across levels of gene expression, metabolic flux, leaf level photosynthesis, in order to address the question, can we simulate soybean photosynthetic response to elevated atmospheric CO2 at multiple scales? So unfortunately, I don't have time to go into the details of the project, but I want to share with you the two major outcomes from this study. The first is that we were able to create a robust simulation where we accurately captured, observed the field response of soybean to elevated CO2. We were then able to use the integrated model to identify specific genes that we predict are regulating those observed phenotypes. And a major bottleneck for us is transformation, being able to transform crops so that we can test our predictions in our crop of interest in the field. We are now scaling up our model integration to the canopy level. And to do that, we are working with a visualization team to build 3D representations of soybean that can interact with a virtual environment to predict whole canopy light capture and microclimate with spatial resolution. We're using the simulated light capture throughout the canopy to predict whole canopy carbon assimilation, and then we can feed that light capture data into the photosynthesis models. And so now that we have this environment, we can test out different canopy architectures to identify idiotypes with improved photosynthetic efficiency. Finally, I want to share some of the strategies that we have used in the Crops in Silico project to engage the community. First, to increase our user base, we're developing a user-friendly interface to simplify the way that folks interact with the Yggdrasil framework. Second, we have an annual meeting and hackathon, and this is free to attend. And not only is it free, but if you look at this picture right here, we also give you cake. And this is a really important opportunity for us to interact directly with the community, where we can provide hands-on training with the framework, and we can immediately gain feedback on what we're doing right, what we're doing wrong, and what we can do to improve or expand that list of capabilities and implementations. Then we went even further. We developed a new journal called In Silico Plants because we're super creative. And in this journal, the goal was that it would be a home for anything related to plant modeling, plant computational biology, and also a place to publish software and tools that are interesting and useful to the community. To bring this back to the Plant Cell Atlas, I see myself as a PCA stakeholder. I'm really excited about this effort, and I hope that one of the outcomes is the further centralization of data and knowledge that I can use to advance and accelerate our modeling efforts. We're the most limited by available data, and it takes the most time to hunt it down or determine if it's things that we need to generate ourselves. So how about you? Which category of stakeholder do you identify with, and what do you want from the PCA? Who do you think are the beneficiaries or should be the beneficiaries of the PCA? And how do you think that the PCA should encourage community buy-in? So this is just a little extra food for thought during our discussion sessions, which I'm really looking forward to. And I want to briefly acknowledge all of those collaborators of mine in the Crops in Silico project, which includes my colleagues at the University of Illinois, specifically Steve Long, our partners at the National Center for Supercomputing Applications, especially Dr. Megan Lang, who's the developer of the Yggdrasil framework, and then our co-PIs that are here in the States and abroad, and of course our funding, which comes from the Foundation for Food and Agriculture Research. So I'm looking forward to your questions and our discussion.